Donc, sans plus attendre, aujourd'hui, nous sommes Québec-Océan et très heureux d'accueillir Jennifer Provencher, qui est chercheur scientifique à la division d'écotoxicologie et de la santé de la faune, d'environnement et changement climatique Canada. Jennifer collabore avec des partenaires de l'Arctique depuis l'année polaire internationale de 2007-2008, alors qu'elle était étudiante graduée en visite en Arctique pour la première fois. Son travail porte sur la santé de la faune dans l'Arctique. Elle axe ses recherches sur les contaminants hérités et émergents, ainsi que sur la pollution plastique. Ses travaux sur la pollution plastique dans l'Arctique remontent aux années 2000 et son équipe de recherche s'efforce d'explorer le devenir et les effets de la pollution plastique sur les 3 côtes du Canada. Actuellement, elle dirige un projet à long terme sur les contaminants des oiseaux de mer dans le cadre du programme de lutte contre les contaminants dans le Nord. Et elle est également coprésidente du groupe d'experts sur les déchets et les microplastiques pour le Canada dans le cadre du programme de surveillance et d'évaluation de l'Arctique. So, Jennifer, welcome. And now, this is your turn.
Merci, Astrid. Thank you, everyone. It's lovely to be here today. I apologize. I usually try to present my slides en français and speak in English, but this winter got away from me a little bit, so I do apologize for my unilingual presentation today. I'm going to talk about some of the work that I've been doing over the last little bit. I am going to turn off my camera during my presentation just because I'm competing with my daughter who's doing online school currently. So, but I'm happy to be here, and I look forward to questions. So I'm going to start my presentation kind of with a little bit of a definition of wildlife health. It is not necessarily intuitive for everyone who doesn't kind of work in the field. Wildlife health, so our group generally, specifically, at Environment Canada, we work on a number of issues. So we do work on ingested plastic pollution, and this is just a picture of a Laysan albatross. We also work on die-off, so pathogens. Some of you might be aware that there's an outbreak of avian influenza currently occurring in Eastern North America that we're tracking. I also do quite a bit of work on oil pollution. There's a group that kind of does more acute oil pollution. So this is an example of a gannet that's covered in oil, but we're doing a lot more sublethal oil pollution currently. We also think a lot about kind of plastic entanglement in my group. And so this is where they're not necessarily ingested, but this macro or mega plastics, they get entangled. Again, related to plastics, but more on the fishery side. We do a lot of work on fisheries bycatch. And then, of course, we look at harvest and harvest levels. So there are other people in the group that kind of focus more on these bottom ones, but my program thinks more about the ingested plastic pollutions, pathogens, and then again the sublethal levels of oil and what that means for bird populations. And so just as a general thing, I'm going to focus exclusively mostly on plastic pollution today. I'm not going to touch on the other kind of series of our program. And so just as a summary, what is wildlife health, especially in this time of a pandemic, where we're studying zoonoses, right? The COVID comes from wildlife and has infected people. Where our research really focuses on is if your habitat is intact, so you're not having a habitat issue and you're sustainably harvested, but you're still having a conservation issue or health issue, that's where you come out as the wildlife health. So it can cover parasites, plastics, pathogens, contaminants, and that's kind of where we situate ourselves. So within the conservation world, but habitat is kind of a different group, harvest is a different group, and we're kind of left with all the other pieces. And so some of the health issues that you might be aware of is certainly white nose syndrome in bats. So that's a health issue, not a habitat issue. And then for those of you who are familiar with kind of the Western landscape, is wood bison. And so we have been doing a lot more work on TB, tuberculosis, brucellosis, and contaminants in wood bison. And wood bison in particular is interesting because it's one of the few species in Canada that has an imminent threat order for 2 of the herds, which is where we focus our work on with Parks Canada and local Indigenous partners. So today I'm going to talk about plastic pollution and specifically plastic pollution in wildlife. And one of the things that you have to know about plastic pollution from the very beginning is that size matters. And so depending on what size class you're talking about, different wildlife can be exposed to them. And so we have these kind of macroplastics, and that's what we see on like the beach litter side. You can go all up to like megaplastics, which is, lost and discarded fishing gear. We also have meso and microplastics. The center kind of size class is what the most animals ingest, and most animals kind of were worried about, at least from a wildlife perspective. This is where, and the definition of microplastics is not standard. It is different depending on what group and what context you're thinking about, but this is really generally in the kind of 1 millimeter and over size. And then we go down into these ultrafine plastics or lower. So we've been calling either the kind of 1 micron to 1 millimeter is either an ultrafine plastic, or sometimes it's called a small microplastic. And then anything under 1 millimeter is called nanoplastic. So again, that is not standard. So this slide, you'll notice, is a little confusing because there's overlapping size ranges. That is on purpose. It is a confusing nomenclature, but the important part that it does matter, and it matters to wildlife in particular when we start assessing how they're affected by plastic pollution. So my lab in particular, we focus on 3 types of plastic wildlife interactions. The first one I'm going to talk about is ingestion and accumulation, and this is what we've been doing the longest, and globally the community has been thinking about the longest. There's a second part where we think about a lot more recently, whereas the seabirds as vectors as plastic. And this is an important one and has actually come into play in international regulation recently. And so we're thinking a lot more about this, how birds, migratory birds, are moving plastics around the environment. And then we are doing quite a bit of work in relation to that on plastics as a vector for contaminants. And there's an interplay between these two. So seabirds as vectors of plastics, who are then vectors for contaminants. And so we'll talk a little bit about this today. So for many of you, probably some of the first images you saw of birds and plastic was by a photographer. His name's Chris Jordan, and he took these really remarkable pictures of Laysan albatross chicks out on the Hawaiian islands. And so these are young birds that have parents who forage in the Pacific garbage patch, kind of in the North Pacific, and are fed plastics from a very young age and essentially starve to death on the island. And so the plastic fills them up, and they don't have any kind of food or nutrition from it, and they starve, and you can see the results. And so they die, and then the carcasses decompose and the plastics are exposed. So this was a starting point for a lot of people thinking about plastic pollution in birds. And as I started my master's, actually during the international pool year, as mentioned, I was tasked with doing a diet study, and what I had to shift to really quickly was learning about marine debris as I started finding plastics in the birds that were collected by Inuit hunters in the Arctic. And so a really quick primer is that marine plastic or marine debris is complex. It's not all from a single source. There are these industrial pellets or nurdles, as they're called, and these are kind of the raw plastic material. So plastic is a source of, or it comes from petroleum products, fossil fuels, they get formed into these little pellets. These are the pellets that are shipped all over the world from refineries. And then once they're kind of at the factories, they get formed into all kinds of things, which is what we call user plastics. And these user plastics include, our cell phones that we're all, sitting and using, and our, chairs and computers, bottles, toys, all the things that we kind of run through the consumer's hand. Collectively, between these two groups, user plastics and industrial plastics, it's been estimated by the U.S. Environmental Protection Agency that about 20 billion pounds of plastics enter the ocean every year. And again, this is the problem for birds because it actually ends up in their stomachs. They end up ingesting it. And so just to give you a bit of an estimate, that's about a dump truck of plastics entering the ocean every minute. And so just since that we started, so in the last 11 minutes, you can imagine, 11 dump trucks, or the equivalent of 11 dump trucks, kind of backing up and dumping that plastic into the ocean. So it's happening at an extremely fast rate. So, we've had a lot of attention to plastics in the last couple of years, but it's not a new thing for wildlife biologists, and certainly for those who study in certain parts of the world where plastics accumulate. And so we did this a couple of years ago now, but the trajectory hasn't changed, where we actually documented debris being in animals and plastics in animals, plastics dating back to the 60s, debris in general, anthropogenic litter, garbage back to the 40s in fish, turtles, mammals, and seabirds. And while fish are, taxonomically quite numerical on the planet, there's lots of fish. It's actually the seabird literature here that has been a vast majority of what we know about plastic pollution over the last, 30, 40 years. The others are kind of coming along. There's a lot more mammals, and fish have really taken off. There's a huge amount of fish work now. But from a kind of a long-term, standardized, harmonized approach, it's really seabirds that have given us the base knowledge on plastic pollution in wildlife. And again, this has to do with the types of studies and groups that study seabirds, the fact that you can access them at colonies in large numbers, and the history of studying seabirds goes back a long way. And there's actually a report published in 1838 of a Wilson's storm petrel having a candlestick inside its stomach. And so, in part, this has to do with a very long history of kind of bird observation and bird studies that have gone on. And so this is where a lot of my work is based in, is understanding these historical patterns as we move into a time where plastic pollution is being reduced in terms of release into the environment. And as we bring policies in, what does that actually mean from a bird perspective? Is it going down? So when I started, again, in my graduate work, one of the things that was really striking is that the northern fulmar monitoring in the North Sea was really the only place in the world where plastics were regularly monitored, and they were done via seabirds. And so, again, dating back to the 70s and the 90s, plastic pollution was on the policy agenda. It's not new. It's not a new problem, and it's not a new policy. There were several different policies put into place that had to do with plastic pollution, specifically in European waters, that really set a lot of the early monitoring of seabirds into place. And so part of the 1992 work that was done was actually naming the northern fulmar, which is a species related to the albatross in the northern hemisphere, and very specifically laying out the number of birds, what the levels in these birds should be, and what the time period is. And I know this is a lot of text, but I put it there because it's very directly taken from the policy documents. And so it lays out your sample size, your sample locations, your test years, what those levels should be. And this is the type of kind of science policy, monitoring policy interaction, which sets very clear grounds, very clear purposes for monitoring. And when you put it into a policy document, it kind of like writing it in stone, at least for a little while. And so, where we can inform these types of discussions as scientists is really to inform these critical policy decisions. And my colleagues in the Netherlands and in Europe who have worked on this have been very strong advocates for some of the science, evidence-based policies. So, of course, we have northern fulmars in Canada. We have northern fulmars on all 3 of our coasts. They mostly breed in Nunavut and Nunatsiavut, so less so in Nunatsiavut, mostly in Nunavut, but they can be found in all 3 coastlines. And so when we started really thinking more about plastic pollution in Canada, and comparing to other regions, specifically the North Sea and Europe, we had a focus on fulmars. And so this is a picture of fulmar breeding up on, I believe this is Cape Vera, way up here at the top of the kind of breeding zone. And this is an example of some of the plastic pieces that we found. You can see that there are industrial pieces. These are industrial nurdles in the picture, as well as these broken shards, which are all user plastics. This one in particular is very identifiable as a bottle lid. It's hard to see in this picture, but there are grooves on the side here that are, the grip for opening the bottle. And so we've been sampling from these 5 locations opportunistically for the most part for the last 15, almost 20 years now. And we've certainly found some patterns that exist. And so this is where I'm actually going to ask you all to do some thinking and guessing. And so we've got 4 sites that we do, fairly regular multi-year sampling at: Vancouver Island, Prince Leopold Island, which is up here in Lancaster Sound, the Labrador Sea off the coast of Nunatsiavut, and then Sable Island is a small island off the coast of Nova Scotia. And so what I'd like you to do is think about what we've talked about so far, what you know from just watching media and other things. And I'd like you to put those 4 sites in order of how much plastic pollution is in the fulmars sampled from these sites. So do you think Vancouver Island is the most? Do you think Prince Leopold Island is the most? And you can just put that right into the chat. And so, what site do you think has the greatest amount of plastic? And which site do you think has the least amount of plastics? And I'm just going to give everyone a few minutes as you type in, and I'll try to follow along. Okay, so I see a few answers: Labrador Sea and Vancouver. So you got to tell me which one you think is the lowest and which one do you think is the highest. So I see PLI, Labrador Sea, Vancouver, Sable. Is that lowest to highest or highest to lowest? So I got Vancouver Island as the highest from a few. Prince Leopold Island is a high one there. So a few guesses. I'm gonna see if there's any more coming in. So Labrador is low, Sable is low as a few guesses. So just while a few other people are typing in, I will tell you that this is a little tricky. I've done this presentation to quite a few people, including our past Minister of the Environment for ECCC, and she also did not get it all right. So it is not a straightforward answer. So we've got Sable, Vancouver, Labrador, PLI. Okay, so one of the first things, I'll show you the answers. So one of the first things is that Sable Island actually has the heaviest plastics. So from a weight perspective, this is our number 1. So the birds on Sable Island have the most weight pieces of plastics. Whereas Vancouver Island, which most people guess as having the highest levels, they actually have lighter loads of plastic, but they have more numerous pieces. And it's really hard to kind of tease this apart. We go by weight, mostly because plastics can actually fragment inside birds, and sometimes you have 10 pieces, but they're all the same kind of plastic with the same color. So, potentially it's one piece that was eaten, but then it broke in their belly. So it can be really complex on how we kind of cite that. But we think from a health perspective or a condition perspective, it's probably more important to be on the weight side. Prince Leopold Island had the least amount of plastics. It's a very small amount of plastics, and the Labrador Sea is intermediate. And so these are the patterns that we've kind of thought about and seen over time. And so it really does tell us that we have a latitudinal gradient. So as we move north on both of our shorelines, we see this. And we see this a little bit clearer in this graph. So we've got a couple of different sites, but these sites are in the western North Atlantic, so Devon Island, Prince Leopold. And then as we move south in our western North Atlantic, we got these higher numbers. The same thing happens in Europe in the eastern North Atlantic. From Svalbard, as you move south along towards the English Channel, you get these higher levels. And then we have fewer sites in the North Pacific, but it's the same gradient. And this red line here is our policy targets, our ecological quality objectives set in the North Sea policy documents. And what's really interesting is when I started working on this, we found plastics in the Arctic birds, and I thought, oh, even in the Arctic, they're really, not clean, right? They're having plastic ingestion. And my colleagues from Europe looked at this and went, oh, this is so great. They're like below the ecological quality objective. And so this work has kind of continued on as we look at these patterns, and our European colleagues actually look at this as it is possible. So if the clean, relatively pristine Arctic birds can have this lower level, then it is an achievable policy target, which is really important for them. But just really quickly, birds are not birds. And so northern fulmars, we have lots of studies. They have, this relatively high frequency of ingestion. So we know that about 85% of our Arctic birds, Arctic fulmars, have plastic in them. But when we look at colonies that have multiple species, so other species such as black-legged kittiwakes, thick-bill murres, black guillemots, at the same colony at the same time, we see much lower levels. So we know that the plastic ingestion actually relates to foraging strategy. So it matters what you eat and where you eat it. So these surface feeders have higher levels of plastics, and then the divers are very low, in this case, 0. And so it really is these species. It's not just about plastic ingestion being in the water, it's about where in the water. So plastic floats for the most part. And so it sits at that surface water interface. And this is exactly where these surface feeders are eating. So we've also done a number of gull studies, and this is a study that a student of mine, Sahar Saif, did a few years ago, and we've followed up a few times. But the diversity of litter in some of these bird species is really diverse. And so this picture just shows that, again, birds are not birds. It's not just plastic, but we've got aluminum foil, we've got rope, we've got drywall and chipboard, other things in here. So this is some of the stuff that we're tracking. It's just not plastics. It's just not straight up plastics. It's often these kind of mixed media. And so we looked at 3 gull species that were all hanging out together, primarily at dumps in Newfoundland, and just looking at, when they're exposed to all kinds of debris, what are the types that they're ingesting? And this has real implications for contaminants, which we'll talk a little bit later. And then, of course, being at the center in the middle of a pandemic, working in wildlife health, when we're, studying COVID, which is from animals moving to humans, and then also in the middle of the pandemic causing more plastic pollution than ever before via PPE discarded into the environment. We've got this kind of nexus of what some of the work that we have been doing versus, both tracking COVID in animals, but then also tracking PPE COVID-related garbage in animals as well. And so we have these studies where we're actually looking at COVID pollution in the environment. And then we have these gulls where we've got kind of pre-COVID samples, early COVID samples, and then deep COVID samples. And so we actually have teams out currently who are doing some collections as well. So we're trying to figure out if when we change the type of plastic or litter pollution, do we actually see changes in the ingested plastics? And this is work by Carrie Shooten, who's a PhD student of mine at Guelph University. kind of shifting gears a little bit, we've also been thinking a lot about how birds can be vectors for plastic pollution. And so this is work that we started to do a couple years ago with the Sululit Area Co-Management Committee, who is the co-management community for the Akpayat and Kakuluit National Wildlife Areas on Baffin Island. And so they were really interested in, so we've got plastics, but where is it going? Is it going into the environment? And so we actually started looking at these kind of poop samples, or these pre-poop samples down kind of right before the birds excrete, and looking for plastic. So normally we would look in their stomach, which is what this is kind of we're used to seeing. But here we started looking at their kind of poop samples, or their, just before they're going to poop samples. And we started looking at microfibers and ultrafine plastics there. And what we were hoping was that, like, the poop samples would be a useful indicator of plastic pollution. So if they had plastics in their stomach, we would find microplastics in their poop. And unfortunately, this is a graph of the data. It just doesn't work out that way. And so when you have microplastics in the stomach, you can have zero plastics in the poop, and then vice versa. You can have plastics in the poop and nothing in the stomachs. And so it didn't work out, but it started this other line of questions where we know that the birds have plastic in their poop. And so we actually did a co-sampling kind of trip in 2018 with the Sululit Area Co-Management Committee, and actually started to look at the two species that we got, northern fulmars, and what their microplastics in poop look like, and their thick bill murs. And then we were really interested in, what the anthropogenic kind of polymer fibers were. So we did Madeleine Bourdage, who worked on this project. She, took the plastics all the way through to FTIR, and then she did some back of the envelope calculations. So if these birds on these colonies have plastic in their poop, how many pieces are they kind of pooping out a year? And so it's counterintuitive a little bit, but the murs have less plastic in their stomach, but they poop a lot on their colonies. And so you can see a little bit here, they're just kind of, colonies are covered in poop, and they're really dense nesters. And so it's been estimated that 45 million pieces are kind of deposited on the colony every year. The fulmars, although they have more plastic in their stomach, they poop less, and they're less dense on the colonies. And so you can see there's a bird here, there's another bird here. They're very sparse in comparison. So we estimate that there are only about 3 million pieces of plastics pooped out onto the colonies every year. And this is of particular importance to the Sululit Area Co-Management Committee as we start to think about, the health of the colonies, the health of the environment around the colonies that they co-manage with Environment Canada. And so we took this the next step to really think about, could the seabirds, like for chemical contaminants, be creating a halo of contaminants or a halo of microplastics around the colony. And Bonnie Hamilton did this paper, again, in partnership with the previous study, where we actually looked at microplastics in the air around the colonies and the sediments around the colonies. So you can imagine there's the bird colonies that we sampled, and then we sampled in the water, we sampled in blue mussels, atmospheric deposition, and then sediments. And the take-home messages to this question of, like, is there a halo of microplastics? The answer is maybe. We had a small number of sites at one location, and it looks like atmospheric deposition. There's microplastics in a lot of the air samples, so we think there's probably some very strong currents that are taking the plastics in a lot of different directions. So there's not a clear signal. But in the sediments, I think if we explore this a little bit more across a couple of different colonies, we might see this actually start to be more of a pattern. It's not significant in this, but we kind of see a trend. So we've got more plastics near the colony. As you move away from the colony, you've got lower and lower levels. But again, this can be complex because of air and water patterns in the region. So we're continuing to work with the Sululit committee on, what their science and monitoring plan is looking like for the national wildlife areas. And that's a big role for me in particular, as a government scientist. I do lots of science for a lot of different reasons, but some of our science and a lot of my program is focused on these co-management committees and carrying out what the science and monitoring they want to do within those national wildlife areas. So the last piece I'm going to talk about is the plastics as a vector for chemical contaminants. And so for a long time, we've thought about the kind of physical effects. You can get blockage or false satiation, so you feel full. I did a lot of this work under the Northern Contaminants Program and supported by Neopar in collaboration with some projects that Max Liberon had. And so we've done kind of thought about what are the chemicals that are potentially leaching out of the birds. And this is where more and more of kind of our work is leaning. And again, I did a postdoc with Acadia and supported by the Weston Foundation on this that we're still really exploring. There's a lot of data that came out of it and we're still kind of crunching through it. But one of the things that we have spent a lot of time thinking about is, so again, just like a bird is not a bird, it's not a bird. Plastics is not plastics, it's not plastics. And so we actually took pieces of plastics that were sorted as fragments and we put them through an FTIR. And so you actually start to get a higher resolution of the type of plastic. So polyethylene was a big one, nylon, olefin, and you can see there's a whole whack of things here. And this data is we're building on. We're getting better. It's not always the beginning was not always accurate, but as more and more people will do this and more libraries come on board, we're getting much better at figuring it out. But the point being is that there are a lot of different types of plastic, and that has a lot to do with the contaminants that they might carry. And so again, we talk about wildlife and plastic pollution and plastic ingestion. And when I started working on this, a friend told me, she's like, I was telling her that I was opening up birds and finding plastics. And she kind of looked at me and she was like, yeah, but like, I think my 2-year-old probably has plastics in its stomach. Like, does it matter? Like, who cares? Like, probably most things have plastic in it. And I think the kind of really important answer to that is we all probably, have plastics that we have food come in contact with, that we might accidentally ingest or consume. But for the most part, our plastics are food grade plastics. So they go through a very specific, examination for leakage of contaminants. Whereas the plastic debris that the wildlife are taking in ingestion are not food grade. So they're a huge cocktail of chemicals and polymers that are not kind of tested for food or contact by kind of digestive processes. And so I think this is a major difference. So we have plastics that are, it's unavoidable. There's, we can't avoid plastics in our massive food processing system. But that type of plastics is controlled, whereas the plastics in the environment is not. It's all types of plastics. And so we've looked at a couple of different suites of plastics. This is one that we've looked at in particular in PCBs. And so we did see different kind of PCB congeners and plastics versus the liver of the birds, but it is dominated by a few different groups, which are pretty standard. And we came, I think, slowly coming to the conclusion that for these legacy contaminants like PCBs, probably plastics are not a significant vector for them because their PCBs, other legacy contaminants, are so present in the food and the prey items that plastics are not kind of an additional significant factor. And so, and this kind of changes when we start thinking about plastic associated or plastic derived contaminants. And so this is actually a study I did with Jia Liu at UCAR, who's an excellent chemist. And we started looking at not just legacy contaminants, but these really plastic specific contaminants. And so this is a group of antioxidants that we looked at, and we looked at them in birds, eggs, and bird livers. And so these are kittiwakes. So if you remember, kittiwakes had about 10% ingested plastics, whereas fulmars had about 90% ingested plastics. And my prediction was that we thought fulmars were going to have much higher levels of these plastic additives as compared to kittiwakes. And this just wasn't the case. And so we don't have a lot of kind of information that, ingestion leads to higher levels of the plastic. But I think more importantly is this, it seems small, but this little egg bar here. And so we find that these plasticizers, these antioxidants, transfer to eggs in both the fulmars and the kittiwakes. And so this is this is a really important thing if you are thinking about kind of health development. So this maternal transfer is present, which is really important and something that we've been thinking a lot more about recently. And we did birds, but we also did seals. And so one of the things that we've been thinking about is, there are certain suites that we find more in birds, and then other suites of contaminants that we've been finding more in seals. And UV328 in particular, which we found in seals and fulmars, is now under consideration by the Stockholm Convention. And so there's that interaction where birds are being kind of confirmed as vectors of plastic to the Arctic. And then those plastics are the vector for some of these contaminants like UV328. And so currently in the Stockholm Convention, they're assessing UV328, and part of that is long range transport, which has implicated migratory birds and plastics in general. And so that is kind of moving along in the background as we start thinking about these international conventions. But interestingly, we were finding these plastic additives in seals, and it really led to the question of what about plastic ingestion in seals? And so I actually had a student, again, Morten Bordage, who looked at a bunch of samples. So it's 142 seal stomachs from Nunavut, collected all by Inuit harvesters. And what was interesting is she looked at plastics over 425 microns, which is kind of the limit of what you can do when you've got historic samples that weren't purposely collected for microplastics. And she didn't find any plastics. There was no plastic pollution whatsoever in any of these stomachs, again, over 425 microns. So, that's about a half a millimeter. We found lots of things. We found lots of rocks. We found lots of parasitic worms, lots of fish, lots and lots of zooplankton, but no plastics. And so this really, I think, is an important part, because you can have birds or you can have animals that ingest plastics, but don't have necessarily high levels of plasticizers across the board. But you can also have animals that have low plastic ingestion, but have plastic additives. And so from this work, we're not thinking that seals are important from a physical plastic perspective, but because they do have plastic additives and they're harvested, that there are important kind of contaminant and country food questions to be considered. And so we've repeated this study, actually. So lots of people say, okay, well, 425, how, maybe you missed a whole bunch. And so we actually repeated this study. Alex Jardine is doing this work where we've looked at samples from Sachs Harbor. And again, we went all the way down to 80 microns, and no plastics were found in any of the samples that were kind of accounted for by the blanks. And so again, it points to the fact that marine mammals may not have a lot of exposure to plastic pollution, particularly in the north. This work has then spawned on, of course, others and other interests. And so we've actually been actively working with, I think it's 5 communities in Nunavut, Snarawak, Naujat, Coral Harbor, Iqaluit, and Pinkerton, collecting walrus. For those of you who work in the north, walrus is a very commonly harvested species. And so this is actually a picture of the walrus harvest and sharing in Iqaluit, and I was there a few years ago. And so the walruses are caught out on the boats, they're tested for trichinella, and then once they're negative, they're put out and people can come down and get the meat. And so we've been collecting the stomachs with the H2O support and looking at them. And again, we've gone down to 80 microns, and there's no piece of plastics found in any of the 28 samples that we've looked at so far. And so this is a good news story. But again, it does kind of challenge this idea of plastic ingestion is going to be ubiquitous across all species. It really depends on where the species live, what they eat. And so the question of vulnerability and susceptibility in these animals. And so I'm just going to kind of leave us on the last little thing is the Canadian Science Plastic Agenda. So for those of you who are kind of aren't deep in this world, this is an exercise that ECCC undertook a few years ago. There was a 3-year funding window for plastics that's just coming to an end this March. And we're currently sitting with a renewal request, a budget request with Treasury Board as we speak. And again, I'm not, for those of you who don't necessarily look deeply at the ministerial mandate levels, for those of you who kind of work or strive to work at the government science policy interface, I encourage you to do so. And if you go back and look at the ECCC mandate letter, you'll actually see, I think it's 8 or 9 points, all point to plastic pollution and plastic science. And so, likely we're looking at a renewal for plastics funding in Canada for the next 5 years. And we're kind of sitting and working with partners currently to see what direction that will go and how that will look across the landscape and where the priorities are. So we've done, a lot of focus on the past has been in marine. And so we're really looking forward to some of the other pieces. So there's 5 pillars in CaPSA. You can go online and look at it a little bit more. But the 2 that we kind of focus on in my work and drives my work is really the detection, characterization of the environment, and then of course the impacts on wildlife, human health, and the environment. And so this is going to be continuing on in the years to come. And so in support of that, and I'll just point this out, is some of the most kind of detailed work that has been done on kind of the assessment and monitoring has been done by AMAP, and I'm the co-chair, as mentioned, of the Litter and Microplastics Group. And so there's a, with most Arctic Council staff, there's kind of a 4-page summary with overview. We've put out a monitoring plan, which is kind of a 24-page version of what all the countries in the Arctic should be doing to monitor litter microplastics. And then there's a very comprehensive 266-page technical guideline. And this is the how, where, and when kind of guide to do microplastics and litter work in the Arctic. And it covers 11 different environmental compartments where we have kind of worked with experts from across the Pan-Arctic region to think about how and, how to measure, how to report in order to work towards, widespread harmonized reporting of litter and microplastics. So I think I'll leave it there, and I'm happy to take any questions.
Okay, thank you so much, Jennifer, for this wonderful presentation. So I will start with a quick question. I'm quite new to the plastic world in the ocean. And is there any project to limit the release of plastic in the ocean in Canada?
Yeah, so that's a big part of what environment and climate change is doing. We have a whole plastics division, and so they sit within the environmental protection branch, and they do a lot of work on estimating what the emissions are, what sectors they come from, how to minimize, how to, what we say is close the circle, a circular life for plastics so they stay in the system. And, there's lots of programs that are kind of looking at even things like recycling. So across Canada, there's not kind of one system for recycling. And so there's many groups and much work supported on, how to keep plastics in the plastic system and reduce the release of the environment. And so where kind of the science policy interface happens is that they're thinking about this from a policy perspective, and part of my job with partners is to, survey the environment, survey wildlife, and figure out what the plastics are. And so that's where they then can go and, look at what the best policy tools are based on evidence from what the biggest sources are. And so just as an example, you can imagine that the plastic, most people are very aware of plastic straws or plastic bags as being a problem. And there are policy tools to focus on those. But the question always is that the biggest source of plastics to the environment? And so we do a lot of work both back and forth with those working on policy to reduce emissions, to focus efforts on the actual emissions of concern.
Okay, thank you. Is there any question? Gabrielle?
Yes, so thanks, Jennifer, for the talk. It was very interesting. So I was wondering, is the amount of plastic found in the Arctic is higher than what is found in the Antarctic?
Yeah, great question. I don't know if we have a good measure on that. So there are sampling cruises actually in the Antarctic right now who are doing plastic pollution sampling. But the work between the Arctic and the Antarctic, I have to say, generally is not very well coordinated. And so contaminants are a great example of that. Our Arctic program, generally, we know a lot about contaminants, and in the Antarctic, they've had a very different focus, and contaminants work generally in the Antarctic is pretty sparse. It's pretty few and far between, and there's multiple reasons for that. And so the amount of plastic pollution work out of the Antarctic is also, like contaminants, pretty few and far between. And so it's really difficult to compare. But one example is there are some water studies that have been done in the Antarctic. And we've done water studies in the Arctic, and what is unfortunate is that they weren't done in a harmonized way, and so they're basically not comparable. And we've written kind of a lot about that, and so plastics, as you all know, probably are kind of a hot topic in kind of public science, popular science. And so there's a lot of people who have kind of jumped on that bandwagon and tried to, do sampling. But the problem is, unlike a lot of other kind of community science or citizen science, is that everything is made of plastics. And so if you don't have the proper field lengths and the proper lab lengths, it basically means that the samples are contaminated by like everything that you're wearing. There was a recent study even that showed most of the plastics in water sampling were from the boat it was sampling from. And so it's a real challenge. So all of that AMAP work that I showed at the end is really about how do we get there? How do we get to samples that are comparable, that are done in the same way, that are harmonized? And then we'll actually be able to start, because right now, we can't even really do a lot of comparisons in Arctic waters. Seabirds are actually one of the few, compartments where we have harmonized methods, whereas we don't have harmonized methods for most others. So seabirds and shorelines of beaches have harmonized methods, and everything else, we're still really trying to push people to use harmonized methods for comparison. So it's a great question, and I unfortunately don't have an answer, and we probably won't for several years.
Okay, thanks.
She?
Hi, Jen.
Hi.
Hi, very nice talk. Thank you. I would like to ask you if you are running any program to measure the toxicities of plastics on the seabird or other species?
Yeah, we don't have any kind of we don't have any captive studies. We do have a number of studies where we've tried to look at the kind of co-contamination. In seabirds, it's particularly hard because we don't really have any captive studies. And there's a lot of other contaminants. And so that's one of our challenges, is we can look at kind of correlation between health metrics, but there's a lot of other contaminants that are all happening at the same time. So kind of to battle that, or to kind of account for that, we actually have a study on captive mallards that we are starting up this spring, where we'll be doing exposures to plastic additives, in mallards that are captive, and we can control one metric or one variable at a time. So we're doing that, and that's very, bird related, and mallards are a great, great model species for that. And the other thing that we're doing is we have an experiment. I didn't talk about it today at all, but we have an experiment in the experimental lakes area. And so most of you are probably familiar with the ELA, where I think it goes back to the 60s, where they have lakes that are by legislation, we can add things to and see how that changes at a lake level. And so at the ELA, I'm part of a large project led by Chelsea Rockman, where we're actually adding plastics to a lake at the large scale level and looking at additive and plastic effects on zooplankton and fish. And then we have some swallows in there as well. So we're trying to get at that in a couple of different ways. But it is a problem in wild seabirds because there's so many other variables that are changing that we just can't account for.
Okay, thank you.
Philippe?
Thank you for your presentation. Really, really interesting. Just have one question. At some point, you talk about the work you did, excellent work you did with Hamilton in 2021. And you mentioned the word mussel at some point, and you know that I'm an ecologist, so I was trying to get more info for that. So can you give us a little bit more detail about what you find, etc.? So you just tease me on that. Thanks.
No, I'm so sorry. We that's the one piece that we haven't published on, and I always laugh when people pick up on that. I should have known you would have picked up on it. Mussels and microplastics is really complex. They do ingest microplastics. They have been argued for monitors, and maybe it's a longer conversation we can have, but they only ingest a size fraction. So what they figured out is if you give microplastics to mussels, they like them, but they sort them, and so they don't ingest the small pieces, and they reject the larger microplastics, and they really sample this one size class of plastic, and so that's what you find in them. And so we've been trying to think about how we can use them, mussels, as monitors of microplastic in the national wildlife areas, because they're great for community sampling. They can be done really easily if you can find them, kind of where they would be. And so mussels and microplastics are really complex, but they do ingest them. And again, from a human health perspective, they have big implications, right? Because they're consumed whole. And so, seabirds, even though they're harvested, no one eats the gastrointestinal tract, right? They eat other tissues, whereas things like mussels and clams, the scallops, they ingest microplastics, and then we eat them all. And so they've got huge human health perspective implications. So it's a great question, and it's there's an entire talk, but we actually just did a book chapter that I could send you if you're interested in more about mussels and microplastics.
Yes, please. I will love that. And just for the subject, there's a really old paper where they use endoscope, and for surgery, but they put that inside mussel, inside clam, inside scallop, and they look at the size selection of particles. That's really cool because you can see it live. Anyway, that's more geeky stuff for me, but yeah.
Totally. But yeah, so they do the exact same thing with microplastics. They size sort them.
Yeah. And they use some specific size micropellet to test that, how they select the size. So I can send you, that's a quite old paper, but they use an endoscope. Cool. Yeah. And a nice video.
Very cool.
Sorry, really geeky here. Sorry.
It's great.
We maybe have time for a last quick question. Felix?
Okay, so thank you for the presentation. It was really interesting. I was more interested about maybe how do you, because you've been talking a lot about like field work, and I was wondering, how do you pick your partners, and how do you organize your teams? And yeah, that was maybe something I was curious about.
Yeah, so it's complex and it's different for every community. So a lot of the work that we do, so as a government scientist, one of my jobs is to support the area co-management committees. And so Environment Canada has national wildlife areas, migratory bird sanctuaries. These are established protected areas. And so part of my work as a research scientist for the government is to support these area co-management committees. And so there are kind of two ways that I pick partners specifically on the ground. A lot of it is not so much picking partners, but actually working with established area co-management committees. So I don't pick to work with them or not, right? They are established, they are community-based, they're community selected, and I go to their meetings, and I often refer to as they give me homework, right? And then I go away and do it, and I come back and we make a plan together. So I'm a little bit different than some of my academic colleagues who do pick and choose who they work with. I don't pick and choose who I work with. I work with established partners who are community-based. And so we have long-term monitoring programs. Some of my programs I've inherited, they've been going on for 40 years, and I work with those partners who are already at the table. And because they're tied to specific sites, tied to specific protected areas, and tied to specific committees that are legally bound, that's who I work with. And so it's a little bit different, but then I do have other work where I do, I would say have a little bit more flex in where we go. And I often work with, so there's two ways in which I work. So sometimes I'm the lead. So all of the birds projects, a lot of my work is out of Québec-Océan and out of Resolute, out of Pond. And those are places where I have worked for several years. I have existing relationships with. And so again, we explore questions that they're interested in and think about that work, and often I work down the current. So I have projects in Resolute, Pond, Québec-Océan, and then also in Nunatsiavut, Nain, and Postville. But then there's a lot of other work that I don't lead, and so all of the walrus work is with partners at DFO, and so they're on the ground, they're doing their projects, and then when they want to talk about plastic pollution or someone brings up the topic of plastic pollution, they call me, and then I lend a hand to them. But I don't. We do work in Sachs Harbor, we do work in Penguin, but I don't lead there because those are mammal projects. And so I do a lot of work where I support other people who are kind of being asked to do microplastics work, but don't have that expertise. I support them in, again, collecting the samples in a standardized way, making sure that we're asking questions that kind of tie directly to the methods that they can do, whether it's physical plastics or contaminants. And so, again, as a kind of a government scientist, my job is to support other programs. So I lead the fieldwork from the bird perspective, and all that other stuff, fish and mammals, is really I'm loaning my expertise, but I'm not the driver in any of those places. So people have come and said, we really want to work on this, and then we come up with a plan together. Does that answer your question?
Yeah, thank you. It was an interesting way to describe the interaction. Thanks.
Thank you so much for your presentation. Pour ceux qui restent, n'oubliez pas, nous avons notre réunion scientifique annuelle le 31 janvier. Donc, c'est gratuit, c'est virtuel. Et on vous retrouve le 31 tout ensemble. Merci beaucoup. Bonne journée.